In this video, children, we are going to discuss the Calvin cycle in detail. That's the C3 cycle. Okay, so now since you have already watched the first video where I have explained why this cycle is C3 cycle, so you must be knowing that here phosphoglyceric acid is supposed to be formed. Okay, so these are the bases, and according to initially this chapter was there in boards, and according to the boards came to these four were the main headings of Calvin cycle. So straight away these headings are going to give you marks if ever this question is there in your exams, right? This is the first step, carboxylation, right? Second step is a glycolytic reversal, you may call it a reduction, right? Then third one is sugar formation, this is third point. And fourth point is regeneration of RUBP, and you know till now that RUBP is a 5 carbon atom compound, right? So understand that during carboxylation I used a word, fixation of carbon dioxide. But before we mention anything else, let us talk about the hero here. Hero, that means the most important enzyme which is responsible for occurrence of this Calvin cycle by fixing atmospheric CO2, and that enzyme is RUBISCO. Just hold on. That enzyme is RUBISCO. Wait. RUBISCO, right? The full form of RUBISCO is ribulose bisphosphate. That you have to write complete. Ribulose bisphosphate carboxylase. Carboxylase. So as of now you can end up writing RUBISCO because it is fixing atmospheric carbon dioxide, it is performing carboxylation. So RUBISCO, this much full form is fine, but always remember children that this enzyme has two functions. It can work the either way. It can fix atmospheric carbon dioxide, and at times it can fix atmospheric oxygen too. So carboxylation and oxygenation, both functions can be carried out by this enzyme. That's why we call it a hero. And you at that time when it performs oxygenation, at that time you will write it as oxygenase. Just add the word oxygenase too. So it is always more precise to call this enzyme as ribulose bisphosphate carboxylase oxygenase. C for carboxylase, O for oxygenase, because it can perform both the functions, right? So understand that this is the enzyme which is very much important. This is something you have to mention, and then you just mark an arrow. Let me use some other color here. You just have to mark the arrow, and you have to show that fixation of atmospheric CO2 is done. So CO2 is actually fixed in the dark reaction of photosynthesis. But remember that carboxylation of what? So here it is carboxylation of RUBP, right? Here you are having RUBP as the base compound, and this RUBP is 5 carbon atom, and carboxylation of RUBP leads to the formation of first immediate product, which is PGA, phosphoglyceric acid, and I told you it is a 3 carbon atom compound, so 2 molecules of phosphoglyceric acid are formed, right? Then they undergo a series of reduction processes. You call them glycolytic reversal processes, and this is the stage where two things happen. I'm using a red color because things are exhausting here. The first thing is the NADPH. Actually it is a reversal, right? So here this H is going to be removed and it will become NADP plus 1 inorganic phosphate separately, right? This is the first change. And another most important change is the conversion of ATP, because here the energy is exhausted, so ATP is converted into ATP. And remember children, you have to link it up with the previous explanation. These two molecules of ATP are exhausted here, and 2 molecules of NADPH are converted. I mean the H is removed from them, right? So in totality, when you tally it with the bioenergetics, where in totality 1 Kelvin cycle was consuming 3 ATP, so which is the step where 1 more ATP is supposed to be consumed more? I mean here it's 2. NADP is okay, because in total 2 NADPH are actually exhausted during 1 Kelvin cycle. But what about ATP? As of now you have only shown 2 ATP consumed. The third one is consumed during the regeneration of RUBP. So here also 1 molecule of ATP is converted into ADP. This is D. Just hold on. ADP, right? So now your bioenergetics is satisfied. 2 plus 3 total 3 ATP in 1 cycle consumed. And for sugar formation, how many, I mean proper glucose, if you need, how many cycles you need? You need 6 times you need this carbon dioxide to be fixed. So 6 cycles will take place, and in total 6 cycles 18 ATPs and 12 NADPH will be exhausted. Come to this stage, this is a triphosphate stage. That means that when glycolytic reversal occurs, at that time the glycolytic reversal means when this PGA basically it is converted into phosphoglyceraldehyde, then it is converted into the intermediate compounds like pseudoheptulose and ribulose, xylulose and all, and ultimately the sugar is formed. But I mean sugar is not straight away the glucose, so intermediate sugar compounds will be there, and then ultimately it will become C6H12O6, right? And since you know that your base RUBP was exhausted in 1 cycle, for the next cycle, once the sugar is formed, for the next cycle the regeneration of RUBP should be there, so that it is available for the next cycle. So regeneration of RUBP also exhausts this ATP molecule so that it can participate in the next cycle. This is all about dark reaction, Calvin cycle, that's C3 cycle.